The other point which has to be included from the very beginning in your facility design is a contingency plan. So one of the things that you need to take into account when you're designing a facility is what if this equipment fails? What if this supply fails? The more in advance that you solve these things, the less the problem is going to be. If I have an autoclave in my facility, what happens if my autoclave fails?
Hey, smart scientists. Welcome to another episode on the Smart Biotech Scientist podcast. What are the critical aspects we should looking after when we're designing a biotech facility? Well, today we're talking exactly about that with Alfredo Martínez Mogarra. He's a bioprocess engineer with a background in biology and 30 plus years of experience in the bioprocess industry in several companies such as Amgen, Serono, now Merck, and Genzyme, obviously now Sanofi. And he participated in the design, the construction, and the startup of several factories in different countries. He also has a wide experience in process improvement for different molecules. So stay tuned for an insightful conversation. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Alfredo, welcome to the Smart Biotech Scientist podcast.
Thank you very much. Very happy to be here.
It's a pleasure having you on the show today. Alfredo, share something that you believe about bioprocess development that most people disagree with.
I think the main surprise people may have with this is that bioprocess is something easy, and it is not. So normally focus is put on the technology, in the molecular biology, in the discovery, which of course is science and is what makes mankind progress. But nobody usually think in the difficulty associated to move these discoveries to something which is industrial. And being industrial is basic to distribute and to get the benefit of these marvelous discoveries. If you're not industrial, your investigation is reduced and doesn't work very much. I think this is the most surprising thing. People is not counting with these difficulties and consider that, well, things are easier than they are actually.
Yeah, and we're going to get into this a bit more later during our conversation because today we have an exciting topic. We're talking about the biotech facility design. But before we do that, I am curious, Alfredo, how did you get started in biotech and how did you get to do your current role?
Well, it was many years ago that I started, longer than 30. I was a student. I was studying biology. This time it was when the production of recombinant proteins was starting at industrial level. And I remember perfectly, I was with my degree, studying my degree, and I discovered the thing of changing the DNA of bacteria. At the time, it was made in bacteria only, industrially, and making this bacteria grow and producing proteins which were human. And I was so amazed by that. I said, well, this is what I would like to do. And probably for most of people who are hearing or listening to me right now, it's not particularly remarkable. But I'm talking about a country, which is Spain, and a time, which is the 80s, and thinking now that I want to go to Mars, in a plane to Mars, and stay there. So it was more or less the situation. There were no facilities of this kind, and of course, no money to do this kind of things. So I had this. I finished my degree and made a master in microbiology. My department was more focused on industrial microbiology, and I started to do things of this kind there. And by chance, I guess by chance, I don't know how much is the fortune on this. Fortune is okay when you're ready for it. And I was contacted by a laboratory, and Swiss-Italian laboratory at the time, and they were looking for people to work in biotech, and they interviewed me and I joined that company, and this was the beginning. So I started my life in biotech producing epidermal growth factor, or trying to produce epidermal growth factor in bacteria with a fermenter of 100 liters. And I received the fermenter, a set of instructions, and a box of tools to control valves and these kind of things. And this was my beginning. From that, I was more and more interested and doing more and more things, moving to other projects, and things progressively grow. At some point, I had been producing human growth hormone for many years. I was in my country. I was living in my country. It was a comfortable life, but something was calling me to do more things in the profession. I left the company I was working for. I had been working for 15 years, and I started an international career. I joined Genzyme in Belgium. They had a facility to produce monoclonal antibodies. I moved also when I started that, things started to move. I moved to Amgen, and I have been in several companies. Each company offered me something more in terms of position or opportunities. And well, life has taken me through this path. I have been also producing synthetic DNA, which is something very interesting. It looks like proteins, but it's not at all proteins. And currently I'm producing viruses. And of course, together with this activity, I have something very interesting and that I really, really like, which is the Biotech Academy, in which we try to centralize all this know-how and supply some cultural, some educational support to new companies and people doing these kind of things. This is the situation. This is how I have been evolving in a few words.
Wow, well said. It's difficult to summarize what you've done the last 40 years, isn't it? In such a short time.
Not so many, 35.
Yes. But since you've seen pretty much all stages of the biotech industry, the very early stages, and now I'd say the more mature stages, I am curious, Alfredo, what do you believe are the main challenges that biotech scientists often encounter in process development and in the manufacturing of all kinds of biologics?
It's a very nice question. I've been thinking recently about this, and I've made something that probably can make you smile, but it's very much evidencing what's the situation with these things. So I'll give you my example. Me personally, I'm a good amateur cook. I cook, I produce some good stuff. People come home, they enjoy, and I celebrate because I like very much the compliments for my food. And now with this basement that I have this stability, I decide to create a restaurant. And then because I'm good cooking at home, I try to create a restaurant. It's going to be easy because I cook well. This is the horror because I don't know how to shop for the restaurant. I don't know how to organize a restaurant. I don't know how to cook for 30 people and not for 4. And this is a bit what is the shock when very nice project, very smart projects try to go to the industry. They find all these problems. And together with this, the money, because biotech is one of the most expensive industries that we have in our time. No, it's building planes, building boats, and biotechnology are probably the 3 most costly industries that we have. So it's a lot of shock. It's what do we do with this, and how is it possible that cannot we do this? No, you cannot do this with a pipette. You need a hose, these kind of things. And it's very shocking. Particularly remarkable is that the people, when go to the industry, think that, it's a matter of biologists, of chemists, of molecular biologists. And they completely forgot that it is also very much a matter of engineers. And this is basic. So you need your engineers. You need, of course, your scientific expertise, but you need your engineers to make it a reality. And of course, you need your people expert in quality and regulatory affairs, because this is very much conditioning the whole thing. And it's very shocking for people finding these things, which are not found altogether. It's a progression, and also people embarking in this kind of projects needs a lot of resilience because they are going to discover hurdles one after the other, not all together. So it's not an easy journey.
Yeah, smart biotech scientists cooking for a few guests at your house is quite a different ball game than cooking for an entire crowd in a restaurant and building that. So, and this is exactly why we're here today, because I'm excited, Alfredo, to talk about a biotech facility design. So let's start out with the most basic aspects of that. What are the most critical aspects to consider when you're designing a biotech manufacturing facility?
So the very first thing, and this is very important and sounds stupid, but in my experience, most of people do not consider this. You need to know exactly what you want. And it's not about, yeah, no, you love, but it's absolutely relevant. You need to know exactly what you want to do. You want to do this product or this set of products. You need to go to this capacity. You want to service to the market. And if you can know your process, it's even better. Sometimes you cannot, because very often you're building a facility which is generic. It's for different products which are going to come or intend to produce different products, or you're a CMO or whatever. But what you need to have a clear idea is what you can do and what you cannot do. So once you have this idea, you need to establish which are the regulatory requirements that you have to produce this, because this means a series of red lines that you need to respect. Otherwise, your facility will never be authorized. And once you have all this, you have this concept, then you need to develop a series of policies. How are we going to do things? If we are going to do single use, if we are going to make open operations in certain environment, tier classification, what are we going to do with residues, which kind of supplies we are going to need, where are going to be placed, and then what the possibility of getting. You need to have all this picture, which is not glamorous because it's not related with the biotech or the molecular biology, but it's absolutely basic on which you need to build your process. So once you have this, you need to make a URS, a user requirement. You have to express in the document what are the things that you expect to see in your facility. And also you need to imagine what are you going to automate in your facility. So all this together gives you a series of documents. And this is something which has to be created by the core team, by the people who are in the process, who have the initiative, who has the project. And then is when the engineers enter. You give these requirements to a group of engineers, and they will take the space available. Maybe it's a brand new construction. Maybe you're using an existing building, whatever. And they need to adapt all these needs, all these limitations, and supply these needs to the space that you have. And this essentially is a very collaborative work. You need engineers, and you need also your internal team to make this continuous evaluation. And then is the time to market, which is always pressure and a problem. Normally, all these facilities, because you put money, when you build a facility, you put money, and you're not producing nothing. And this is a very uncomfortable situation for investors. So you have a lot of pressure for the time to market to start the profitability of your facility. So this is very relevant. You need a lot of coordination, and you need that people make it as much in advance as possible, and always having a lot of attention on what are you building to make sure that you are not forgetting anything. Very much contact with the regulatory authorities. This is very helpful as well. Present your project, asking the authorities, do you agree with this, do you agree with that? Do you think it's going to be acceptable? They will never tell you if it's okay or not. And the meetings are not binding meetings or promises of anything, but you will have much more probabilities of having something. And at the day, don't forget that the regulatory authorities of your country have two goals. First goal of a regulatory authority is to protect the patient. And this is why they exist. The second one, which is not so clear, but is to help the national industry. So they are not going to do as much as possible to create you difficulties. They are going to do as much as possible to help you in saving these difficulties. So it's a good communication with regulators is interesting. But this thing is planning, knowing the process, knowing what you want, and making planning and review this planning and have a proper design. And thinking a lot before starting construction. The more you think, the more you save.
And how do these initial steps, when you are still in the conception phase, in the design phase of your facility, differ between a greenfield project and a brownfield project? Are they very different, or are both of the steps very similar?
Well, greenfield is letting you doing things better, because you can put all the imagination that your money allows, which is important as well. But it's easier at the time of implementing solutions and creating a new facility where you have the functionality, you have the ergonomy, all these things. When there is something which is pre-built, you may have more difficulties. Maybe your efficiency at the end of the project is not as good. But the point is that it's much cheaper when you have something already built normally. You save the investment in the ground, you save the investment of the construction.
Now, let's look at the process side, because when you're designing a new facility, obviously you want to produce something in that new site. So what are the common misconceptions about transitioning from the bench scale to the industrial scale?
There is one very typical, and I have seen it in a few places. People think in bioreactors. And the bigger your bioreactor, the more beautiful and intense your facility. And forget completely that this bioreactor one day finishes and you need to take the 2,000 or the 5,000 or the 20,000 liters and put them somewhere to process. And they forget about the dimensions of the downstream. And this is very typical. So something important to take into account. You need to have an idea of your full process. You need to consider how many people you're going to need, because something which is usually skipped is, well, I need this, so many operators, I'm going to work in 3 shifts, blah, blah, blah. Okay, where are going to be the dressing rooms or your operators? Have you ever thought of that? Well, anywhere. No, anywhere not. You need to determine a place and you need to make people feel comfortable, because people feeling comfortable work much better than people who are thinking in leaving that horrible place. So it's all this stuff, which is, we go to the glamorous part, which is a bioreactor, where you have the cells, where you take the samples and make the magic. But you need to take into consideration all these other things around. You need to solve your problem of different supplies. You need to evaluate which are going to be your consumptions. These engineers do very well. You need to solve things like how many square meters of magazine I'm going to need to maintain a production. And this is not that easy, because you need to know the supplies that you need are something which arrives immediately or is something which is taking time to come, because this is helping you in designing this thing. And this point is very relevant. These kind of things are also to take into consideration. If you need a storage tank, if you need to cool things. The other point which has to be included from the very beginning in your facility design is a contingency plan. So one of the things that you need to take into account when you're designing a facility is, what if this equipment fails? What if this supply fails? The more in advance that you solve these things, the less the problem is going to be. If I have an autoclave in my facility, what happens if my autoclave fails? Well, I need to stop my activity unless I have this a second unit or I have this security about something. No? If you're installing normally bioreactors, you don't have a spare bioreactor. But you need to understand what's going to be the impact of a failure in your bioreactor. If this is something that you can solve with your maintenance, you need to make sure that your maintenance is going to be in the situation of solving this problem. And you need to have clear idea of which are the spare parts that you may need and have, I don't know, maybe a couple of months of delivery time. So you need to have provision of these kind of things in your magazine. It's a lot. It's very multidisciplinary, and that's a lot of aspects that normally people do not suspect. They are focused in the process, in making the process, growing the cells, purifying things, and not even thinking in the impact of the size of things.
So why are the operational aspects so different between the large scale and the lab scale? Is that just because of the scale, or does anything else create these huge differences?
The first thing is because of the scale. I can give you an example. We had a process in a company, for one of those I've worked for, which included a reactor feed of glucose. And this feed of glucose was starting in a very extremely concentrated solution. So concentrated that it was in the limit of the glucose solubility. And see that the glucose is something really soluble, but it was in the limit, in the edge. And the only thing to have it well dissolved in a reasonable period of time was heating the solution at 50 degrees or something like this. And if you're working on the bench, having a glucose solution of a few milliliters at 50 degrees is not at all a problem. But if you have 200 or 300 liters of liquid at 50 or 60 degrees, it's quite a problem. It's a safety problem. So this is one part. Is the scale? Yeah, is the scale is a problem. The other thing is how things manifest then, because you can manage a few milliliters with a clamp and don't touch it. But if you have this thing, you need probably a tank. And if you need a tank, you need the space to move your tank. And you need to develop the procedures to move this, and you have to consider this in the design. So this is why it's important to know which is the product that you're going to produce and which are your limitations. And alternatively, if you have a process of this nature and you cannot face this kind of practical problems, you have always the possibility of making some development and try to find a shortcut to avoid this situation that you cannot sustain with your facility. And this is taking longer. and maybe some impact in regulatory.
Yeah, this glucose feed is a great example of something that seems minor at small scale, but is a huge issue at large scale. So it's time very well invested up front to think about these issues and then find solutions. And this brings me to the next point, because when you're building a facility, when you're scaling up, there are a lot of risks involved. How do you mitigate these risks?
Well, the risk analysis is something very relevant and very much underlined by the regulatory agencies. The best thing is teamwork, evaluating what can go wrong, try to find alternative plans, and having always a plan B for everything. In extreme cases, for example, a manufacturing facility is something where you have to supply from two different independent sources of electric power. And first time I heard that, I said, well, this is crazy. No, it is not. It was a facility in my past again. I don't want to mention names. There was a good, reliable electric power supply until one summer, the heat was forcing everyone at home to connect the air conditioning. And the supply of power failed in summer. And we had to do miracles to maintain that because there was no alternative power supply. We had to do miracles to keep that, and we had people working in the manufacturing areas at 35 degrees, which is a nightmare. So that was. So having plan Bs and then making sure that if something goes wrong, because practically everything can go wrong, you have an alternative plan. Even for the regulatory agencies, if you're producing something particular, like, I don't know, DNA, for the DNA, we had this, no? If you are supplying synthetic DNA in your facility, it's not a common thing. There are not so many places. If you have a minor catastrophe in your place, like an earthquake, something like this, that interrupts your activity, how are you going to supply the market? Because this is the obligation of the regulatory authorities. They need to support the patient safety, and a shortcut in supply may mean this problem for patients. Not at the end of the chain, there are people suffering. So one of the questions that you even have to put on the table is, if I have a fire, if I have a flood, if I have an earthquake, whatever, and my facility has to stop for 1, 2, 3 months, how are we going to supply the market? So even these things you need to make sure that you have considered. And this will open you the doors to do things.
Hey, smart biotech scientists. Have you been enjoying our conversation with Alfredo Martínez Mogarra? Tell us what you think about today's episode by leaving a review. Alfredo Martínez Mogarra is a bioprocess engineer with 30 plus years of experience in the industry, and he participated in the design and construction and startup of several factories in different countries. On Thursday, we're continuing our conversation about the factory design, about scale-up, and how to manage all these different stakeholders. Stay tuned for an insightful conversation coming up this Thursday. And perhaps you are in a startup project as well, or you are scaling up your process, and you have a lot of questions and you don't know what the critical factors are. I want to offer you a free 30-minute assessment to get started and to help you prepare your project correctly. So use the link down in the show notes, and I'll see you on the call. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.